invitation. And I apologize for any possible English mistake or mispronunciation of some words. But I will try my best. I'll talk slowly and carefully. So the title of the presentation is Ceratocystis wilt on eucalyptus and mango tree caused by Ceratocystis fimbriata sensulatum. So Brazil and Australia, we have a lot of common interests and also climate. So I think we are something like up here, okay? So it hits about this location here in Brazil, which is quite hot too and warm and nice beaches. In here we can go to the beach. I think in Australia we cannot go so easily because you have a lot of crocodiles and all kinds of wild animals, but we have a lot of common interest in climate. Anyway, we are 13 hours away time, okay? But Rafael and myself, we already got used to the new time, so now we are like the good Australians. So I work at the Federal University of Vicosa. We are located in a small town in the Minas Gerais state, which is a southeast state. And our town, it's here in Vicosa, in the southeast state, southeast part of the state as well. It's about 350 from Rio de Janeiro, a city that most of you have heard about, mainly about probably about Carnival, and it's a nice city, and so that's probably the most famous city in Brazil, Rio de Janeiro. But we are in land; we don't have sea. Okay, so. So that's a little bit about the town. Vicosa or Vicosa is a small town. It's mainly a university town, very small, about 73,000 people. It's a very small population. Out of that, almost 30 are students. Okay, so almost half of the town are students. Our climate is tropical altitude, which is quite comfortable, and we are at almost 648 meters altitude. We have a rainy summer, and the maximum temperature is 28 degrees, and the dry winter it's about 8 degrees, but sometimes you get a little bit colder. Especially these last days have been quite cold in Vicosa. That's an overview of the campus. I work here at the Biotech Institute. And that's one of the oldest buildings that you have. Currently, it's main use for administration. And that's an overview of a night from the university side. Last year we had about 17,000 students, most undergrads, about 2,000 undergrad, 2,000.5 undergraduate students, 2,000.7 graduate students, and 1,000 and something non-degree. 647 high school students, and currently we have 65 undergraduate courses, mainly in agriculture. We are very strong in agriculture, especially agronomy, forestry, veterinary science, and most recently we also have medical science, engineering, civil engineering, several social courses. So The university is expanding quite a lot in the last few years. And we have 45 master programs and 23 PhD programs. We have a very strong international relations, including with Australia. I myself, we had a program, a project with Australia since long before the Myrtle Rust came in here. So we are very keen in having international collaboration. But currently we have more than 90 academic agreements with more than 30 countries. We have 200 students abroad and 240 students. The Brazilian government has presently a program to send students abroad. It's called Research Without Borders. It's either for undergraduate students and for graduate students. So it's a recent program that we are sending our people abroad to get more international knowledge, and so it's a very interesting program. And we have also a couple of students from other countries. The Brazilian government has a fellowship program for students coming from South America and Africa. So it's in most of our universities are free. We don't have to pay at the universities, okay? And so it's something like maybe different from other countries, okay? We have also some students from North America, from USA, undergraduate students. Well, let's talk about eucalyptus and mango. Okay. So first I would like to talk about eucalyptus, because eucalyptus is my favorite tree. I love eucalyptus. And the experience that we have got on eucalyptus, we are applying on disease of eucalyptus, we are applying to other crops as well, and like mango that I'm going to talk about today. So anyway, my favorite tree is eucalyptus. So eucalyptus in tropical sites have a very high growth rate. Our current mean annual increment in industrialized managed euc forests in Brazil is about 40 cubic meters per hectare per year in average. So it's a good growth rate. I'm not sure about Australia, but in some places in Australia maybe you may have 15 or 20 cubic meters per hectare per year. In other countries like Uruguay that I'm aware, it's about 15 to 20. Portugal is 12. So we have maybe the best growth rate of eucalyptus in the world. Our rotation age is about 7 years, but currently we are trying to reduce this by 5 or 6, the rotation age. But at a 7-year rotation age, we have over 300 cubic meters per hectare. So it's quite a lot. But in some sites and for some clones, we can reach up to 80 to 90 cubic meters per hectare per year. And we have also some information, some experimental plots. It can reach up to 150 cubic meters per hectare per year. So we have a very good tree improvement program. So we expect to have better growth almost every year. Currently, we have an integration of eucalyptus and different crops and cattle and horses. It's a very good model for the small growers because I don't know if you have an idea, but in some countries like Brazil, there are some misconceptions about eucalyptus. So many people think that eucalyptus take too much water from the soil and take too much nutrient of the soil. So sometimes we have some protests against the eucalyptus. So now, and then also they say that we are expanding our plantation too much and taking the land from producing food and so on. So this is a very good model for the small growers. They can integrate their own crop with wood, so wood production. So they can increase their profits doing this integration. So it's a very interesting program. So we can integrate this with rice, for example. So in the first year, they plant eucalyptus and rice, and they have to leave a space between close to the plants, about 3 meters wide, without any competition. And then they plant the rice in between, and then they harvest rice and plant soybean, okay? And then they harvest soybean, and they seed grass, which is very good grass for cattle, for growing cattle, for beef or dairy. And this kind of system, it's very comfortable for the animals because you don't have much light, and the grass is more suitable. It's richer in protein content, and it's greener, so the cattle likes a lot. So it's a very good system, the integration of crop and cattle. We can also integrate with horses. Here, an example of eucalyptus and peanuts, eucalyptus and castor bean. Castor bean is a plant used for produce oil, to extract oil for bioenergy, okay? Biodiesel. You can also integrate with sunflower. I don't know if you have sunflower here. You do? Okay. Our forest plantation in Brazil is currently about a little bit over 7 million hectares of plantation, and most of it is eucalyptus plantations. So we have currently over 5 million hectares of eucalyptus, which makes 71% of the total forest plantation. And secondly, we have pines, about 1.5 million hectares of pine plantations, mainly Eucalyptus Pinus ellioti, I don't know how to pronounce correctly, and some Pinus taeda as well. And we have also other forest species like African mahogany. You have also Toona ciliata, which is native here, and some native species like Suetena macrophylla, which is close to African mahogany, and other few native species. But as you know, we have most exotic plantations. And maybe as here, the exotics behave much better than the native species. So for us to add up, for example, rubber tree is much better outside Brazil, like in Malaysia, than in Brazil itself, because we have a lot of natural enemies, and it grows much better out of the range where it's nature than elsewhere. So why eucalyptus? So eucalyptus has a multi-use purpose wood. So we use eucalyptus for different uses in Brazil, but mainly for charcoal production. And charcoal is used in the steel industry. And currently we have a new technology of producing charcoal, which the smoke is burned and it generates energy also. Okay, so we don't have pollution. So all smokes that would come out from the the ovens, it's burned in the special area here. So it's quite a good, modern, good technology, recently developed, and it was developed in my university, this technology, and now it's being used by several companies. So it's very interesting. We have also used eucalyptus for, most recently, for solid wood, like fence poles and furniture. But the main use of eucalyptus is for pulp and paper. Like here we have some rustic furnitures like this, and some nice piece of furniture, beautiful wood. And the treated poles and fancy poles or electric poles, because we cannot harvest our native vegetation or native forest, so we have to plant. And eucalyptus is the best source of wood for different purposes. And to me, this is the future. It's present, and the future is timber to use as very good timber. Some species are very good for timber, and there is increasing interest in producing eucalyptus for timber in Brazil. You have a lot of investment from outside Brazil investing in this forest for producing timber. So it's very interesting. So as a consequence of the expansion of the eucalyptus plantations, we have had several disease outbreaks, and like Cryphonecter canker or Chrysopus cubensis canker, Erythrithrus aulic canker, Ceratocystis wilt, Houstonia wilt, Ervinia wilt, several leaf diseases caused by bacterial and fungus, and rust. That's the topic that we are going to talk on Monday. Okay. Today we are going to talk about the Ceratocystis wilt. So that's the topic of today's presentation is Ceratocystis wilt on eucalyptus and on mango. Okay. Ceratocystis fimbriata sensulato. It's called sensulato because we have recently many colleagues have described new cryptic species, many based on molecular markers. So Ceratocystis fimbriata sensulatum has a very broad host range. It's very similar to Puccinia psidiae. Okay? It affects mango, sweet potato, colocasia, melina, black acacia, single coil, and other single black water. All right? Rubber tree, fig, kiwi, eucalyptus, etc. So we have several hosts for this pathogen. This is on Melina arborea. This plant species was used at the beginning in the north of Brazil by Ludwig. Ludwig was an American that established a plantation in Pará state, and he brought from Japan a pulp in a boat, and he established a pulp mill on the river. So it's quite an interesting enterprise that he did. So it was used mainly for pulp and paper production at the beginning, but the plant was hit by Ceratocystis fimbriata. So it also affects colocation. I haven't seen colocation here, but you may have colocation. Okay. It also affects fig. Fig is a plant used to produce fruits for Christmas. Okay. And recently we found it on kiwi, on kiwi plants in the south of Brazil. And according to our data, we believe that the fungus is also native in this area. Yeah. And eucalyptus, okay? So on eucalyptus, you can see the fungus can kill the plant and also cause a canker. But the most important diagnostic feature of the disease is the wood discoloration starts from the center of the trunk, okay, like this. So it's quite easy to identify. That's the main symptom for this disease. And of course, it kills the plants when it's affected. Sometimes we can see some wilting, and these strips, black strips along the time when you made a longitudinal section. And not common, but sometimes we have also wood borer attack. And many people think that this pathogen is spread, it needs a vector to spread the disease, but it's not true. It has a vector. Some insects can spread the fungus, but most of the infection come from the soil. And eventually, the plants are attacked by the insects, and the frass coming out from these holes can spread the fungus. Okay, so, but of course, the insect can also spread the fungus, but it's not a vector itself. Okay, only. So here you can see the vascular occlusion by the fungal structures. And in the field, sometimes we see some dead plants like this. But nature teaches us how to do things, okay? We have here a clone at the same age of this. This is highly resistant, and this highly susceptible to the pathogen. So this is a clue how to control the disease. Okay? This is by resistance. So what we have done is screening for resistance our eucalyptus. And eucalyptus is a very good plant to work with because we have plenty of variability. We have a lot of variability, then we can select resistant genotypes, and we can clone. Different from most of other plants, differently, in eucalyptus we can clone. So it's a very good tool that we have. This screening we have done at a Clonar disease resistance company, a base technological company incubated at the Federal University of Vicosa. Actually, this company, we established this company. Rafael is one of the partners because the companies, we had a high demand for a screening for resistance because the companies in Brazil, they produce a lot of clones every year. Maybe every company, maybe 100 clones, 200 clones. And before planting, they want to know if they are resistant or not. So to do this screening, we established these facilities, and it's currently done at Clonar. So this is a view of the for plant growth and inoculation facilities. Okay, these are the research team of Clonar. We are three together, the partners of the company. These are our research fellows. In Brazil, we have some incentive from the federal government, and the federal government is is giving some money, some grant to do this kind of activity, okay? And so we have some fellowships from the federal government. These are research fellows, and these are our employees. So that's an overview of screening for Ceratocystis wilt. We inoculate, we have to inject the spore suspension in the plants. And it takes us about 60 days. We have to grow the plants for about 60 days, and we inoculate them, and 60 days later, we evaluate for resistance. So the evaluation can be done by incidence. It's yes or no, if the plant is dead or if it's not. But also we measure the lesion size, and to compare the different clones. We also use some controls, resistant controls and susceptible controls, just to make sure that it's how our clones fit in between them. So here, for example, you have a highly resistant clone and a highly susceptible clone. This disease was described in the late 90s, and shortly after we tested several clones, and we could see that we have a great variability among the commercial clones. So for this company, for example, what they did, we screened the main commercial clones, and currently they avoid to plant the susceptible or high susceptible ones, and they plant only the resistant clones. We have screened almost 900 clones, and about half of them, 54%, are resistant, and 45% susceptible. Okay, so Ceratocystis on mango. Okay, so because we had some relative experience, not great, some relative experience with eucalyptus wilt, we are invited by Vale. It's a company. Vale is a company established in Oman to try to use the same technology we use on eucalyptus for mango. Okay, so how the Omani people could manage the disease in Oman. So we started a project similar what we have done on eucalyptus in Brazil. Actually, this project is a bit broader. We have several people involved, and we are also studying a disease in Oman. It's a witch broom caused by a phytoplasma on citrus, phytoplasma on citrus. Okay, and they also have date palm, but date palm is mostly insect, so we are not involved in that. So this big project is Ceratocystis and also on mango, and a phytoplasma witch broom on citrus, on lime, actually on lime. But I'm going to talk only about the Ceratocystis. Okay. So here we have the main mango producing countries. India is the first one. Most mango production in the world is India, followed by China, by China, and then Thailand. Okay, Pakistan, Mexico, Indonesia, and Brazil is the seventh country. Okay, so that's the main mango producing countries in the world. I don't know where Australia is here, but maybe a little bit less, most for local consumption. Okay, probably. So in Brazil, our the culture of mango in Brazil is mainly in the state of Bahia and Pernambuco and São Paulo are the main state producing mango. And the production of mango in Brazil has increased quite a lot in the last few years. Okay. And let's talk about the Ceratocystis in Oman. First, mango in Oman is an important local culture, local crop. Okay, it's mainly for local consumption. And in 2004, the mango production in Oman surpassed 8,100 tons. And in 1998, they have the first record of Ceratocystis wilt. They also call this the sudden death. Sudden death. And in one year later, 60% of the trees were affected, and in the region of Albatinã. Okay. And the main symptoms of this disease is the death of lateral branches, like this, and some gummosis, gum production, and some strips that dark strips in the trunk, the stem, and differently from eucalyptus. In mango, we find a lot of wood borers. Okay, so that's a close up of the symptoms. Okay, you can see better here. And it's, as I mentioned, it's quite common to see the holes in the trunk caused by the bark beetle. And the worst symptom is the death of the plant, as you can see here. Our previous work has shown that the fungus Ceratocystis from mango and eucalyptus are very close together. You have a group, a separate group here clustered together, eucalyptus and mango, and they have another group here, mango and eucalyptus, and you have a third group here, mainly from mango in the northeast of Rio de Janeiro. Ferreira was one of my students, and we have done this work in cooperation with Thomas Harrington from Iowa State University. He's our collaborator. And we have some students going to his lab, and he has also come to Brazil several times. So he's my main partner in this project. So what this girl showed us was that we think that our Ceratocystis from mango and eucalyptus are very close, and they are in the same group, and they can also interbreed. Okay, so you can get fertile progenies crossing the eucalyptus from mango and the fungus isolated from mango and from eucalyptus. I have currently one student doing a more deep work in Tom's lab. So maybe we may have cryptic new species here, but so far we think that we have on species with differences in genetically different. There is other research like Mike Wingfield's group from South Africa. He thinks that the fungus from Brazil, he named as Ceratocystis mangivora and Ceratocystis mangicola, and in Pakistan he called this as Ceratocystis manginecans. These are the most common in Brazil, And at the moment, we are calling as Ceratocystis fimbriata sensulatum. But according to the papers published by the Mike's group, they belong to different species. So as for eucalyptus, we strongly believe that the control of the disease is by resistance, screening for resistance. So we can see here a previous work done by a colleague in Brazil to have a great variability for resistance. We have several highly resistant varieties and some highly susceptible varieties. So we strongly believe that that's the way of control the disease. So our hypothesis for this project, because this is an ongoing project with the Omani colleagues, that Ceratocystis fimbriata population differ in genetic and physiological variability, and the mango varieties differ in resistance to mango wilt caused by Ceratocystis fimbriata, and new mango varieties with new allelic combination can be obtained. So we also have a program for breeding mango varieties, and we think that we can get new varieties, new resistant varieties by breeding. So we expect that from this project to have a knowledge of the genetic structure of Ceratocystis fimbriata population mango trees in Brazil, in Oman in Brazil. And so far we have some clues that the fungus from Oman and Pakistan are the same, but different from Brazil. And we are trying to track down the pathogen movement. Where does it come from? So, so far, the population of the fungus from Oman and Pakistan are very close together, but different from Brazil. Also, we expect determination of the phylogenetic relationship between the pathogen population in the two countries, determination of the genetic base of the resistance for mango wilt, that's a genetic program that we have, and determination of the origin of C. fimbriata population in Oman, and determination of the virulence variability among different pathogen genotypes. So that's what we expect from this project. It's an ongoing project, and we started it about 2 years ago. So we have already some results. So we did a huge collect of the fungus in Oman, like we did here for rust, for myrtle rust. We just finished. We didn't finish yet, but we're still gonna collect some more in Melbourne. But we did a huge collection of the fungus in Oman, and this one we have been there for. That's Tom Harrington, my colleague, and these are some Abdullah, local colleague, and that's my student Leo, who is doing the working on the genetic variability of the fungus, and this is me, okay, collecting. So in Brazil, we collect the fungus from different places, and the blue circles are locations that we didn't find the fungus, and the red ones that are where we have found the fungus. So that's another view of our collection in Brazil. That's Tom Harrington again. Okay. So we have so far 158 strains. We use the carrot slice to isolate the fungus. This fungus likes the carrots, so we place the wood chips between the carrots, and the fungus grows nicely. So we have obtained 158 isolates so far. And we have also tested the aggressiveness or the variability in aggressiveness of strains of the fungus, and to have a great variability in terms of aggressiveness. So it's very important because we have to select the best strains to do the screening. So because some strains have very low aggressiveness. So we have to select the right ones, the ones that induce more disease on the plants to screen for resistance. So that's one of the, we have screened 40 varieties of mango. And we are using this strain because it's one of the most aggressive ones. Okay, that's inoculation. One of the method that we use is replacing a piece of bark, and then we put the inoculum plug of the pathogen. But we can also, actually for eucalyptus, we use mainly spore suspension. Okay, so, but this can also be done. We developed this technique for eucalyptus canker in the late 70s, many years ago. But we also do it for mango. That's we evaluate the disease. We evaluate the severity of the disease. I mean, we measure the lesion size of the in the plant, and we divide by the height of the plant and multiply by 100. So we have the percentage of severity, of disease severity. That's one of the means. Or you can also just use the size of the lesion. So here we have a great variability of resistance. You have some highly susceptible plants like Aden, several Aden 2H, Aden. I don't know if you are aware of this varieties. Some American varieties, others are Brazilian varieties. But you have some highly resistant varieties like Kent, Uba, and Pravia. It's Pravia? Which one is this? I cannot read. And anyway, we have a great variability for resistance. So that's why we strongly believe that this is one of the main methods for controlling the disease, like eucalyptus. So these are some ongoing studies. So currently we are studying the genetic diversity of Ceratocystis fimbriatus strains collected in Oman, Brazil. And we are also comparing with the fungus from Pakistan. And we recently got some also got some isolates from India in Oman. How do you call Oman? Pomegranate? How do you call it? Yes, yeah. Yeah. In India, they don't have it in mango yet, but they have it. And we are also doing some phylogenetic analysis and population genetics. Of course, resistance, because we have a nice variety of mango. It's very sweet and it's small, but it's very tasty. That's Uba variety. Uba is a town close to Vicosa, so because this variety it's spread in that area, so we are evaluating the resistance of this variety Uba with different strains of the fungus, and we are selecting mango varieties with broad spectrum of resistance for commercial planting in Brazil and Oman. So that's the main objective, is to have resistant plant material to safely plant in Brazil and Oman. So that's our research team. I have several colleagues from my university and also from Sultan Qaboos University in Oman. It's a big group, several students. And this is just to show you some of my, that's Friday afternoon lab activity, as I mentioned that was just before I left Brazil, so maybe the Friday after, maybe I didn't have so many people like this, so maybe just. But actually, we have a good people working; they work hard, and that's my research team. I have currently 5 postdocs, including Rafael. Okay, it's here. Okay, and I have a couple of graduate students, and Patricia is working on myrtle rest, and she's coming to Robert to work with Maureen Glenn on myrtle rest. She's going to stay here for a year in the sandwich type program that we have. And this Leo, who is working on Ceratocystis, he's in Iowa. And so these are PhD students, these are master students, couple of undergraduate students. And in Brazil, we have a very good system. We have fellowships for undergraduate students. So we can, they get some money, and they do some work, they learn, and then we can select the best students for degree, for masters and PhD. So it's a very good system that we have. We have about 25 people working in the lab currently. So that's my technical team. I have also some people. And another thing is that I would like to say that most of the money that comes from my lab comes from the companies. We have a very strong relationships with the forest companies. So about 75% of the grants that I have in the lab come from the forest companies, the private companies. Okay. So these people are hired. I hired them to work in the lab. And this is my technical assistant, Marcia. She's very helpful all the time. And thank you all for the presentation. I